cell. The first thing we're going to talk about is the Golgi apparatus. The Golgi apparatus receives proteins and lipids from the rough ER. Next, we'll talk about the rough and smooth ER. ER stands for endoplasmic reticulum. They are smooth and rough. Protein synthesis occurs in the ribosomes in the rough ER. Ribosomes are where protein synthesis occurs. There are these little green dots right here. Right now, we'll talk about the cytoplasm. The cytoplasm is the cell substance between the cell membrane and the nucleus. We'll also talk about ventricles, the movement of molecules between inside and out of the cell. We will also talk about the central dogma. It is an explanation of the flow of genetic information within biological systems. We will also talk about the nucleus. The nucleus is a part of the cell and contains DNA, which is used for protein synthesis. The cell!
So next, let's take a look at DNA versus RNA. DNA is deoxyribonucleic acid, and it is double-stranded and found in the nucleus. The DNA base pairs include adenine, thymine, cytosine, and guanine. RNA is ribonucleic acid and is single-stranded and found in the nucleus and cytoplasm. The RNA base pairs include adenine, uracil, cytosine, and guanine. Next is transcription. First, the basal transcription factors bind to the promoter, or the Tata box, and then the RNA polymerase binds to the promoter, found near the beginning of the gene. Once bound, RNA polymerase separates the DNA strands, creating the single-stranded template. Next, one strand of the DNA or template strand acts as a template for RNA polymerase, allowing it to build from the 5' to 3' direction. The RNA transcript carries the same information as the coding strand of the DNA, but contains uracil instead of thymine. This is pre-mRNA. Then the 5' cap is added to the beginning, and the 3' poly A tail is ended. Translation. There are 3 stages to translation: initiation, elongation, and termination. In the initiation stage, there are a ribosome, mRNA with instructions for a protein, an initiator tRNA carrying the first amino acid, methionine. First, the tRNA carrying the methionine attaches to the small ribosome subunit, and together they bind to the 5' end of the mRNA by recognizing the 5-GTP cap. They move along the mRNA until they reach the start codon. First round of elongation. tRNA starts at the P site. Next to it, a new codon is exposed called the A site, and that is the landing site for the anticodon who matches the exposed codon. Once the matching tRNA has landed in the A site, the methionine from the first tRNA is transferred onto the second tRNA. Once the peptide bond is formed, the mRNA is pushed through by the ribosome exactly 1 codon. This shift allows the first empty tRNA to exit via the E site. Also, it exposes a new A site, allowing the cycle to start again. Termination occurs when a strip codon enters the A site. So included in translation is aminoacyl tRNA synthetase, which is an enzyme that attaches the appropriate amino acid onto its tRNA. This occurs in the elongation stage. Learn how to read a codon table. First, locate your first letter of your codon using the left side of the table, labeled first letter. Now move to the second letter of your codon. Look at the top of the table where you see the title second letter. Find the second letter and follow it down until it intersects with the first letter from the left side. You should see 4 amino acids. Now, look at the third letter in the codon. Look at the right-hand side for the third letter list. Find the third letter, which will intersect with the box that had 4 other choices. Move your finger from the third letter on the left over to the left, and you should land on the amino acid. Cool. I think that's all I needed to do. Let me check before I stop this recording. Cool. What do we need a codon table for? A codon table is used to translate the mRNA code into the amino acid sequence.